thank you so much for having me here on this DDIP virtual conference. So I am sharing my screen. And so actually I am filling in for my colleague, Dr. Paul O'Shea, CSO of Exemplify Biopharma, who was originally going to talk about a synthetic process for a novel drug candidate. Unfortunately, Paul is unavailable for the talk. So I am filling in for him upon a very short notice. So as you know, my 25 pharmaceutical career, I primarily spent in late stage development, basically from first in human to the final market registration. I don't really have a lot of experience on or interaction with the preclinical drug discovery. So I have been really thinking hard what kind of topics or experience I can share that would be relevant or useful for the audience, for preclinical, for the folks who work in the preclinical space. So as Margaret also mentioned in her presentation, one of the plan is the CMC process development. So this is kind of the most advanced stage. Once your company or your compound, your drug candidate is chosen for further development. So I think understanding FDA requirements and the regulatory guidelines in terms of CMC requirements is really integral part of meeting the milestone necessary for timely and efficient IND submissions. Our company has been working with many kind of biotech companies, academia, as well as the virtual companies who develop a novel treatment in the preclinical discovery or medchem space. And sometimes all of a sudden their compound is past all the successful milestones and suddenly is ready for IND so then they because of lacking experience or understanding of, I guess, FDA requirement, then they kind of come to us and say what is the next step. So I think having this understanding will really help you with drug supply strategy. You make GLP batch, you make a GMP batch in the same batch, you make separate batch. How do you send a proper RFP request for proposal to your CROs and CMOs? What is a proper CMC, which stands for chemistry, manufacture, and control data package? Whether you develop this asset in-house or you change hand to another company. So what is the time and the budget in order to get to the IND stage? And it just really overall help you understand the pathway and the plan for successful IND submission. So today I am going to focus on the chemistry and the quality aspect of the CMC data package. And hopefully this topic is useful and relevant to our audience. So I just want to give some explanation for the abbreviation and acronyms in the CMC world. So IND stands for Investigational New Drug. CMC stands for Chemistry, Manufacturing, and Control. And inevitably, we use ICH guideline, which is International Conference on Harmonization. You heard a lot. It's Q3 and 7, 1A that governs impurity controls, quality, stability, so on and so forth. GMP is good manufacturing practices. GMP can happen in the laboratory, but still it is not GLP, which can be confusing to some of the folks. RFP is request for proposal, RSM is regulatory starting materials. So I like to kind of break down for the IND filing template for drug substance sections. So by breaking down to the individual section and its key content, and I can explain what are the regulatory expectation, what are the quality control focus, what will be a proper I'd say, items or requests in your RFP. So for a IND, the drug substance section, it breaks down to 7 chapters. The first one is general information, including the nomenclature, class number, INN number, your compound code, chemical structure, and molecular weight, and the basic properties, including pKa information, log D, log P, solubility, so on and so forth. Whether your molecule is salt or free acid, free base, whether your salt is a liquid or solid or a conjugate. So including primarily your API definition. So the second part is quite heavy, is the manufacturing process. So manufacturing process, including your CRO, CMOs, your synthetic flow chart, we call it the GMP pocket, start from your regulatory starting materials. It's called RSM. If your synthesis has 18 steps, because you're using a medchem synthesis, you don't necessarily need to include all 18 steps in your filing. You need to define your RSM where you have the control, and the RSM is a whole nother topic, so which will be out of scope for today's discussion. But typically, you include about 3 chemical transformation steps, and could be 1 crystallization or purification step of your drug substance. And you also need to talk about the reagents and solvents used in the manufacturing process. And the third one is about the compound characterization or structural elucidation, which include the 2 parts. The first part is for your compound, structural elucidation, including chemical composition, crystallinity, polymorph, chirality, and chirality control. The second part is about process impurities and the likely impurities that are present in the drug substance, like related substance, residual solvents, metallic or inorganic impurities, as well as genotoxic impurities. So again, including 2 parts for your main compound as well as process impurities. So number 4 is very analytical heavy. It's called control of substance. So that's including your GMP specification, all your analytical release testing procedures, methods, method validation for primarily for non-USP, non-EP methods, such as HPLC methods, gas chromatography methods, that's specific to your compound. And your batch analysis for your GLP tox batch, for your GMP batch, and so on and so forth, as well as justification for the specifications. So chapter 5 is reference standard or reference material. At the early stage, you may not have a reference standard established. You're just using the most pure sample as reference standard. So this is okay. If you do use a reference standard or a purified sample, you need to provide characterization and assign purity, log number, COA. But if you don't have, it's also okay. It will be established as the program continue to develop. So number 6 is about the container closure system or packaging for bulk. Number 7 is about the stability. So stability including stability protocol or stability commitment, stability test data, retest period for the bulk, and sometimes also need to include force degradation studies. At the time of a regular IND, you need to have at least 1 month stability study under long-term condition and accelerated conditions. So, with the time constraint, I'm going to focus on S3 compound characterization and S4 control of drug substance, which is focused on the analytical chemistry, because these information need to be appropriate in your RFP. If you did not set appropriate RFP and in the end, after 6 months or 10 months, you get your drug substance and you find out that they did not really meet the IND requirement, then, you know, it will delay your timeline and you have to probably remake the batch. So that's why this understanding is very important for your planning. So the characterization for small molecule here, so these are for definitive structure elucidation. This is for a scientific understanding. You need to do on a representative sample, on a pure sample, but you only need to do it once for IND, and for the future IND update, unless there is a new form or a new manufacturing process, you don't need to do it again or amend your IND. You don't need to update your IND or IMPD. You only need to establish once. So again, all of the necessary proton NMR, carbon NMR, if you have fluoro on your structure, you need to do fluoro NMR. And also you need to have all the full peak assignment. For exact mass or molecular weight, you need to use high resolution LC-MS or MS-MS to confirm the exact mass. And you need to have peak interpretation of major fragment peaks or add-up peaks. You also need to establish FTIR spectrum and have full vibration peak assignment. For UV spectrum, you need to acquire in the range of 190 to 800 nanometer, especially for EU filing. Some of the UV spectrum is collected from 190 to 400, which is not sufficient. So such as German agency want to see all the way to 700, just to see if there are any observability under ambient light. And if you have a crystalline material API, you need to obtain X-ray powder diffraction. So this is a optional if you have a crystalline drug substance. You also need to present some thermal characterization by TGA or DSC. These are more a physical characterization as a basic property. If you have a chiral compound, so ideally, if you have single crystal for your chiral compound to determine the absolute configuration, that would be great. But a lot of times at early stage, it's difficult to obtain the single crystal. At the minimum, you should have a optical rotation and a proper control for your chiral compound. If you have a salt, chloride salt, sodium salt, you may include a pH titration for the compound or for your compound ion. So as again, this is a scientific structure elucidation is non-binding. You only need to do it once. So the second category, also for S3, is about impurity characterization. So this is very, very important. So impurity characterization is governed by various ICH guidelines. So so I just break down to different categories and what kind of a data package or techniques are commonly used, and what's the degree of data interpretation, how it's going to impact your specification and method validation. So the first one is most important is related substance, which means process impurities, degradation product, byproduct that's introduced from your synthesis. Mainly is in the GMP pocket. So at this stage, it can be residual starting material, residual intermediates, and can be just process impurities. Most companies, they need to identify by LCMS has a proposed structure. Sometimes they have the HPLC retention information. So these will be sufficient for initial IND. And the second category is residual organic solvents, which is governed by Q3C. So in your manufacturer chapter, you do need to list all of the organic solvents used in the GMP process, especially the final solvents that contact your final drug substance, as well as all of the ICH class 2 solvents. So class 3 solvents are considered non-toxic and non-toxic solvents. So class 2 and class 1 are considered more toxic solvents. Class 1, such as benzene, will be prohibited to be used in the GMP manufacturing process of drug substance. Class 2 solvents have mild toxicity and need to be well controlled and well monitored. You do need to test for water content by Fischer titration. You also need to test for elemental impurities or metallic impurity. Could be introduced from your vessel or could be introduced from catalyst you use. The technique we use is ICP-MS. And there's a ICH guideline governing the limits, control limits. The other important is the genotoxic impurity evaluation. So at this stage, you need to use in silico evaluation of your GMP process, starting from the starting material, intermediates, related substance, byproduct solvents. So if you have a structure that is likely to be formed, the structure of impurity likely to be formed, that need to be evaluated. So, and based on the evaluation, you may have to develop analytical testing and the test for the batch, and the batch need to be below. But most time for early stage, a in silico evaluation is sufficient. For the microbial, you need to collect the data, but data may not be included in the initial IND. So for chiral impurities, chiral impurities usually is controlled through process control. You need to have pure chiral starting materials in intermediate, so it can be controlled through process control. As I say, a lot of compounds do not have single crystal at this stage. So if you have HPLC chiral separation, it would be sufficient. If you have a optical rotation for your API, that can be sufficient as well. Polymorph, not necessarily required for initial IND. You can use crystalline form or amorphous. If you have a, if you have to control the form, so that can be set in your internal specification. but not necessarily include in the IND because it's such an early stage. Finally, degradation products that can be discussed in the stability, force degradation, and the process impurities. So again, these are the information that are expected in the chapter 3. So I would say finally, chapter 4 is about the control of substance. This control really means regulatory control for your GMP or clinical batches. So these are binding. So you need to demonstrate methodology, validation of the methodology, as well as your batch quality that successfully meet the specification. Your specification also need to be consistent with ISH guidelines. So here on the right-hand side, I'm showing I would say, generic specification for a small molecule drug substance for oral. So where you see the test methods and acceptance criteria. So for the first one, appearance, so that's straightforward. For identification, you can have FTIR and HPLC retention time. So these are QC friendly tests. As I say, some people file NMRs. As I say, NMR, you just need to do it once in your compound characterization. It's not really QC friendly test, so not necessarily in your specification. So if you have compound, you can have optical rotation or specific rotation here. HPLC assay could be titration, could be QNMR. Typically, it's assay by HPLC. And we also have impurities. So I just list a typical specification here. Total impurity 3%. Qualified impurity, again, based on your safety margin. You can have up to 1% or 1.5%. Unqualified impurity by ISH guideline, maximum 0.15%. For residual organic solvents by gas chromatography, I just listed some example here. These are class 3 solvents. They are usually maximum 5,000 ppm. I'm sorry, methanol is a class 2 solvent, so the limit is tighter, 3,000 ppm. And then I listed the ICP, the elemental impurities here. So class 1 and the class 2A elements, these are mandatory for your drug substance. Whether they are introduced in your process or not, these are mandatory testing. So if you use the palladium catalyst or some in the GMP pocket, you also need to test for that. So you need to check the, again, ISHQ3 guideline to see what are required, but what I listed here is required. So KF, you test for by Carl Fisher, you can have no specification, you just report results. And also, I have seen many cases, a client list a footnote here, the impurity profile of drug substance log released for clinical will be supported by material. It's basically your tox batch or GLP batch used in the preclinical safety assessment studies. So I think, yeah. So I think this is the last slide of my talk. And Exemplify is a CRO located in Cranberry, New Jersey. We offer both consulting and the laboratory services for the IND enabling studies. Thank you for your time and thank you for the opportunity. I'll be happy to answer any questions.
Thank you so much, Yadan, for your presentation. And I just want to make a note that Yadan, Dr. Paulo Shea, John Balesta, and Kelly, if you are interested in discussing any of your drug development projects, you can schedule a meeting with Yadan and her team through our scheduling app, or you can email myself, Sherry, Victor, or our team, and we can always connect you directly with Yadan. So once again, Yadan, thank you again for making this presentation on a short notice.
Thank you, Sam. Thank you for the organizer. Really happy to be here.
You're welcome.